lecturer
Yeah.
from the University of Leicester, and I'm really grateful to be invited by by Emmanuel and Lamis for organising this webinar, and I wanted to try and explore some lessons that I've learnt from a specific problem that I've encountered with antibodies, and maybe there are some lessons for the wider biomedical research community. This work that I'm presenting today is done in collaboration with lots of different institutions, people and places, and I don't for one moment want you guys to think that I'm presenting this as just my work. This is a shared piece of work across lots of different groups. So what I want to talk about today, because I realize not everybody is from biomedical science, I'm going to do a very quick overview of what are antibodies, why are we talking about them, why they're used in research, and also why does this problem matter? Why does it matter to me? Why does it matter to science? Why does it matter to society? And I thought that a good way of explaining that was to talk about a specific patient treatment selection problem that I have in clinic in my other job as a interstitial lung disease doctor. And then I'll introduce three aligned projects that we have that I loosely call better antibodies, better science, better patient outcomes that I think are closely linked. And that looks like a standardized open science quality control collaboration called YCharOS, or Antibody Characterization by Open Science, a stakeholder engagement and coordination plan, which is the Only Good Antibodies community, and then a pilot project that we've only just started where we've got some early data around how we use robust tools to try and help us stratify patients to make better treatment decisions. So I guess the overall objective is to share the approach we've used, talk positively about the progress we've made, but also to highlight where perhaps progress isn't as fast as we would like. What have we learned, and what can we do, and what perhaps can you teach me? And that would be very useful for feedback and discussion at the end. So antibodies. I realize I'm talking to some of you on the call may know more about antibodies than I do. Others of you might only be aware of antibodies because of the news in relation to COVID, for example. But I'm going to go very simple. An antibody is a molecule, a protein that is made by your immune system to detect things and to help you attack infections. So, and COVID-19 was a great example. So your body makes antibodies to fight COVID-19 infection, and lateral flow tests, which some of you may have used to do self-screening, they use antibodies to help show you whether you have COVID-19 or not. And for example, there was a drug made to treat COVID-19, which was also an antibody that attaches to the proteins inside the virus to inactivate it. I can't remember its name at the moment, but it doesn't matter. So in research, in biomedical research, we often use antibodies to find, detect, and label, and sometimes quantify proteins. And that is fundamentally really useful so we can understand the where a protein is and whether it might be an interesting target for a drug, for example. There's lots of different ways of making antibodies, and I'll touch on these a bit later. And those different ways of making an antibody have different reasons for changing the lot-to-lot variability. An analogy I sometimes use when trying to explain antibodies to students and what we're trying to get them to do in research is like finding a needle in a haystack. Now, I apologize if that's a very United Kingdom-centric analogy, but ultimately the problem is not just how good you are at finding those needles, but how big is the haystack, how many needles are in the haystack, and does the haystack also contain fake needles or proteins that look like what the antibody is meant to detect? Um, I think it's worth just very briefly touching on the fact that the type of antibody I'm talking about today are research use antibodies used in biomedical research in assays such as Western blotting, immunoprecipitation, immunofluorescence, flow cytometry, or immunohistochemistry. But there are also other antibodies used in drugs and used in diagnostic tests. And depending on where you are in the world, those have different regulatory frameworks. And the regulatory framework governing the quality of research use antibodies is primarily buyer beware. You are responsible for ensuring that the antibody is performing as you want it to, and if it isn't, you may be able to get your money back, but you certainly can't claim any other associated costs of lost research resources, for example. So to try and make this more concrete, why am I interested in this problem? So I'm going to transport you to one of my clinics in 2010. And I would see a patient who has had a CT scan of their chest and they are breathless. Maybe they have a cough, maybe they've lost some weight. And they want to know, why am I breathless? And back in 2010, one of the diagnoses we were using was this term called cryptogenic fibrosing alveolitis. This is a term that seems to incorporate lots of different types of inflammation and fibrosis, but our understanding has moved on and we no longer use that term. And if someone were to ask me what's going to happen to me, I'd say, well, it depends on your specific diagnosis. It's very variable. I can't really predict what's going to happen to you and what will make it easy. All I can say is what's happened to you in the past is likely to carry on happening to you in the future. Is there treatment and will it work? So as standard for this diagnosis, we were giving immunosuppression with azathioprine, steroids, and N-acetylcysteine. And then in 2012, a group in America did a randomized control trial called the Panther study. It was the Ragu et al. Group, and they actually discovered that this combination of treatment was associated with a worse outcome than placebo in a randomized control trial. And I guess I like to highlight the fact that just because a doctor says something doesn't make it true. Now, obviously, a lot of you will know that very well on this call, but it's worth always revisiting the fact that um, knowledge can be revisited. Things have changed in 24. The progress has been made. We're no longer using that treatment regime. We use an expert panel to make decisions about what your diagnosis is. We call that the MDT, the multidisciplinary team. Unfortunately, we still can't give you any more information about what your specific prognosis is, and that makes it difficult for me to decide whether certain treatments are useful for you. And I still don't know what treatments to give patients per se, although there is a little bit more evidence and new treatments to slow down progressive fibrosis, but I find it hard to identify who is going to have progressive fibrosis. So the MDT is a meeting that sits together of expensive experts who try and make a decision about what the diagnosis is and make a treatment decision based on what do the scans look like, what is the patient like, some blood tests, as well as some other tests in some centers that we don't currently use. And ultimately, this problem highlights the fact that we are using expert consensus in the place of objective, robust criteria. And ultimately, when these meetings discuss the same patient twice, the agreement is only moderate. And there's a critical need to improve our selection of patients for potentially harmful treatments, as well as design better clinical trials. We need good biomarkers and diagnostics. The underlying patient population I'm working with is very heterogeneous. This is just an example of patients admitted in my region with exacerbations or shortness of breath across 6 hospitals, and there were 443 different patients. And yes, one type of ILD, idiopathic pulmonary fibrosis, was the most common, but it still only makes up 1/3 of the total population, and we need robust tools to help us decide what to do for these patients. So the patients are really complicated. So one approach to reducing complexity is to look at model systems. So for example, cells, these are fibroblasts that are thought to be really important in this disease process. And it turns out they're also heterogeneous. I'm not going to explain this data in general in detail. It's from this publication in the British Journal of Pharmacology. But ultimately, we discovered 2 populations of lung fibroblasts, and one were very sensitive to cell death and others were resistant to cell death, and an important disease-causing cytokine transforming growth factor beta 1 caused this the phenotype to switch to the cell death resistant phenotype. And the marker that helped us find out which population was which was a protein called TRPA1. Since we've also discovered in research we've not published yet that important drugs that are used in quite widely, such as steroids, dexamethasone being an example, have the same effect as TGF-beta1, while inflammatory cytokines such as TNF-alpha do the opposite. Is this relevant to disease? I don't know. Could this marker that we've discovered help us decide which patients will benefit from which treatment? You need an anti-inflammatory approach, you need an anti-fibrotic approach. And this is where we get stuck, because actually, although we found an antibody and an assay that worked for flow cytometry, we couldn't find an assay that worked in tissue samples from patients. And actually what we did is we looked at all of the most commonly cited antibodies in research for TRPA1. We produced positive and negative controls, and this is what a clean Western blot signal that for a specific antibody would look like. And unfortunately, only these two antibodies gave these specific signals. And at the time, they'd not been used in any research. And these were the most commonly cited antibodies. Two of them couldn't detect the protein, and one of them detected plenty of other things that could invalidate the results. I was shocked and appalled. I'd applied to the MRC. They'd given me what I thought was a large sum of money to do this research, and I had to go back to them and say, well, actually, some of the underlying data in my application relied on a tool that wasn't fit for purpose. But then I teamed up with Carl Laflamme in Montreal, who was having a similar problem with an entirely different protein called C9orf72. And ultimately, the collaboration led from Montreal led to this publication in eLife, where what we did is some basic quality control experiments of 614 antibodies with using CRISPR-Cas9 to knock out the protein as a control. And we looked at the performance of antibodies in Western blotting, immunoprecipitation, and immunofluorescence across polyclonal, monoclonal, and recombinant antibodies. And we found that there was a very high prevalence of unsuitable research tools, antibodies, and we estimated that maybe each protein of the 65 proteins that were studied, there were maybe 12 published papers without using unsuitable antibodies. And then when we looked at antibodies that had done, for example, because there were too many to look at all of them, we looked at ones that had done badly in IF. We found that there was only assay-specific validation data to show that in about 11% of papers. So the tool probably wasn't fit for purpose, and the publication didn't show any data to say that it was fit for purpose in their specific context. So the problem that we identified, which is contributing to my clinical, not being able to solve my clinical problem, is a much bigger problem. And in some way, shape, or form, it's been recognized for maybe 20, 30 years. However, this piece of work went a long way to quantify how big that problem is. And this feeds into, this is one determinant of the failure to reproduce important findings. And this is a paper, not my own work, Friedman et al., PLOS Biology 2015, looked at how when people try and replicate the key findings from previous biomedical research, how often do they succeed? And this is actually the failure rate. So in the original Amgen study, they tried 53 key experiments and managed and were unable to reproduce, either because they couldn't find out how it was done or because when they did try, they got different results, the vast majority. So if you look at these together and you make a conservative estimate, you would think that about half of research isn't reproducible on the basis of those studies. And ultimately, that's in the this was a I'm afraid this is published. I would characterize it as a back of the envelope kind of calculation. But ultimately, if we assume about half of research is not reproducible, and we take big research budgets like the US used to have, then you can see that's a profoundly large waste of resource. And what this study did was try and break down the reasons that this preclinical research was not reproducible. And it found that biological reagents such as antibodies was one of the most important reasons and reference materials. I deal with both of these in my research, and I'll discuss this a bit later. So ultimately, the reason I'm bothered by this is because I can't get to precision therapeutics for my patients, because this process of going from a hypothesis to a new precision therapeutic is slow, expensive, and risky. And the reason is that there's barriers all over. The tools that we're using are not selective or sensitive enough. Some of our model systems, particularly in my disease area, do not recapitulate the human disease very well. And as I was showing earlier, we have all of these different labels to describe patients, but we don't have robust tools to characterize that heterogeneity in the patients I'm seeing. So we don't have those biomarkers, so we can't develop high quality clinical trials with patients who are all likely to respond to the same therapeutic approach. And sometimes we don't necessarily listen to patients' priorities and experiences in our design of research. And ultimately, the inability to replicate our findings in pharma labs is a massive barrier to drug development. So I'm part of three projects. Like I said, better antibodies, better science, better patient outcomes. And these look to find ways to identify the most possible robust tools, promote the use of those tools by the researchers by making use and choice and use of best practices more feasible, easy, and rewarding, as well as empowering the best reagents towards diagnostics that can help me solve, well, help our community solve patient selection problems. And the ultimate vision is open sharing of trusted data, and we are there. That is happening. Consensus action plans with key stakeholders, that's in progress, and novel diagnostics to help us get the right patient with the right drug at the right time. And this is very much at the early stages. So the first project is kindly funded by the UK Medical Research Council. And what they are doing is working with us to try and develop some frameworks for improving the selection and use of antibodies in research. And what this looks like is developing databases, training materials, applicant guides for funding applications, and author guidelines in partnership with publishers. But ultimately, an important component of this was to say, well, what drives antibody choice and use decisions? And working with a behavioral scientist, Dr. Eva Krokow, who'd previously worked on trying to understand doctors' antibiotic prescribing behavior, we did some focus groups and surveys. And this is a qualitative summary of some of the themes that came up. So when we asked people how they buy an antibody, the things that mattered were the perceived quality or number of citations in the published literature, the reputation of a vendor, as well as other factors such as cost, the availability of free samples, and what was less frequent was the availability of high quality characterization data, and clearly that may reflect an education gap. When we asked them why they felt it was challenging to check that their antibody was performing as they expected or hoped in their specific assays, there was a lack of awareness that the antibody quality could be so low. They were primarily incentivized by publishing hot papers, which means focusing on the interesting hypothesis that they're trying to test, not on checking that their reagents are fit for purpose. And obviously, time and cost, the perception that they shouldn't be the stakeholder who is performing quality control, and ultimately cultural norms and support from both the resources as well as the leadership in labs were considered to be factors. And then the ideas that community generated, and we back this up with larger surveys, was that they felt that awareness and education were key, and they needed more trusted data that they could use to make better antibody purchasing decisions. And they also suggested that publishers and funders might need to support incentives around best practices. I've not talked about all of the themes that came up. Those were the key ones that were backed up by larger surveys. So what are stakeholders already doing? So if we look at some publisher guidance around how you should report your antibody use and what you should report, we took a sample of 30 journals that are very highly cited in biomedical research, and we looked at their submission policies. And about half of them would like you to both submit to include a research identifier, RRID, so that you can uniquely identify which antibody you used, so that other people can replicate your experiment because they know which antibody you used, but also to include validation evidence. And what that means is show that your antibody is doing what it's meant to in your assay system. Now, there's a disconnect here. I showed you earlier only about 11% of papers were including this type of data, and yet over half of the guidance suggests that you should have this type of data. And our hypothesis is this is around the fact that enforcing these guidelines is actually very tricky. So it's okay to set a policy, but to check that the policy is being followed is not so easy in this space at the moment. A number of journals don't actually ask for validation evidence either. When we looked at funder guidelines, there was lots of heterogeneity here. So, for example, the US NIH does ask people to include details of an antibody validation plan and how they are going to identify antibodies. Others felt that ultimately it's the responsibility of learned societies and others to determine scientific practices. And again, there's not a uniform approach, and ultimately some perhaps rightly feel that their scope should not be to enforce tight, specific standards around specific issues. So what we did was we brought together these organizations, which I broadly call representatives of end users of antibodies and research institutions, experts in both the production and validation of antibodies, people who host databases around antibodies, some funding organizations, and some publishers. And the first step was bringing them together in a workshop that was hosted in London at the beginning of last year. And we took to them this data and said, what can we do together as a stakeholder community to try and make better practices in antibody choice and use more possible, easy, and rewarded as a framework for behavioral change effectively that makes behaviors easy to adopt. And the key points from that stakeholder community were we need to increase education and awareness for all stakeholders. And that's part of why I'm really pleased to be invited to talk here. The publishers agreed that actually the gap isn't so much having a policy, it's the ability to make that policy work in practice. And they felt they needed support to be able to do that. And there's different ways that that could look. And maybe the era of AI could be an opportunity here. But for now, our experimental work suggests that that's some way off. We also identified some funding organizations that had dedicated funding streams to enable the discovery and increased uptake of robust tools. Now, those were the NC3Rs and the Michael J. Fox Foundations, and they'd made the decision to invest in that space because they understand the critical reliance upon these tools, not just for reproducibility's sake, but for the sake of the patients and the people that they're ultimately trying to do their work for. And they highlighted YCharOS, antibody characterization by open science, as a key initiative that was making a lot of progress in this space. Um, and what it was doing was working together with the manufacturers to make antibody quality control more efficient, cost-effective, and open. But for now, this initiative doesn't have as much data as we would like. And so one of the things we did was to join this initiative and to start adding data to this space. And that is the Better Antibodies mission, and coming soon here, but ultimately I would suggest look up YCharOS.com, YCH. I'll pop it in the chat later, and you can see standardized wild type knockout antibody validations, and I'll show you a better example in a second. And what this consortium does is it brings together the stakeholders who fund research, the manufacturers who make these tool antibodies, and open dissemination tools. And these stakeholders contribute some resources and state which key proteins they're interested in. These stakeholders provide a little bit of resource, some reference materials, i.e. knockout cell lines, as well as the antibodies. And then a third-party central labs based in McGill in Canada, in Leicester in the UK, and in Cornell in the US perform third-party independent experiments to look at the quality of these reagents. And the experimental data set looks like this. Wild type, WT. So this cell has the protein, KO. This cell doesn't have the protein. And so therefore this antibody is unable to detect a difference, suggesting it's not fit for this specific purpose, while this antibody shows wild type specific signal and none in the knockout. It looks quite simple, but ultimately this is not as easy as you would think, because another problem is the reference materials. Sometimes the knockouts actually do contain truncated or alternative forms of a protein that, without working together with all of these different antibody manufacturers who have antibodies at different epitopes of the protein target, you wouldn't know whether your problem is the antibody or the reference material, whether it becomes a chicken and egg problem that's impossible to solve alone. And ultimately, the credit for bringing these different manufacturers together belongs to YCharOS, and that's led from Canada, and it was the brainchild of Aled Edwards and Peter McPherson. Happy to give you more details of them later. The second experimental pipeline they use is immunoprecipitation, and what this does is it checks an antibody's ability to capture the protein of interest. Now, this particular workflow was of interest to us because if we could develop the correct immunoassay for the right target, we might have a patient stratification tool, but we don't know which antibodies to bother to start with. If we're trying to develop a new ELISA, it's not a small undertaking, it's expensive. So in this data, you have the starting material, the unbound fraction, and the precipitate. And what this does is it assesses the ability of this antibody to concentrate the target. And an antibody that performs well tends to look like this. And so for this target, which happened to be synaptotagmin 1, this one, this one, this one, this one, this one perform reasonably, and the others don't. What you're looking for is concentration here, depletion here, and that's your starting reference material. There's a very similar workflow for immunofluorescence. What we do is we mix wild type cells and knockout cells, best shown here in this 14511 AP, and we then label them with an antibody. And an antibody that performs well, this outline is a knockout cell, and it's not stained at all. And these outlines are wild type cells and they are stained. So this antibody appears to be selective for synaptotagmin 1. And we share this data openly, and I will put into the chat where you can find this data. And we've done about 150 targets so far. This is what the flow cytometry work looks like. So this is a single cell application. These histograms represent populations of cells. I'm really sorry for those of you who don't have good color vision or good color displays, but ultimately the pink histogram here represents knockout. Green is wild type, and this is what an antibody, or this is what an antibody that performs reasonably well should look like. The histogram of the wild type should be further to the right, brighter than the histogram of the knockout, and we use a secondary only control as the background. So this is an ideal antibody. And we share this data openly. Here at Leicester, we've been specializing in accelerating the flow cytometry application. And I guess one of the lessons we've learned from this is for 90% of targets we've studied, we've been able to find a robust tool. So if you work with us, we'll find you something you can use. But in order to achieve this, we had to test an awful lot of antibodies because there's only about a third pass this particular testing procedure. And again, polyclonal antibodies are particularly poor. Monoclonal and recombinants are better. I wouldn't make too much of this difference yet because it's influenced by the specific targets we chose to study. And CD44 is a target with lots of excellent monoclonals that might be skewing this data for now. So I had several reasons for wanting to talk to you. One was to try and hopefully give you the impression that it is possible to identify a problem and start to make progress on it. Not as much progress as I would like, but two was to highlight possibilities for collaboration. We're really eager to find ways of working with the community to help them access the tools they need to do robust research. And the reason I would try and give you for working with us is that we have access to about 60% of global antibody reagent manufacturers. We test 15 to 20 antibodies per target. We're able to solve that chicken and egg problem that I described of, is the problem the antibody or is it the reference material, the knockout or the knockdown? We're able to find you the right tool to be able to study what you need to study. And actually, we also have examples where we are able to work with the manufacturers where we don't find a good tool to develop new tools for you. And ultimately, this makes research more ethical, sustainable. It overcomes those barriers, it makes it more cost effective, and it helps you reach the goal of your research. And ultimately, one of the key goals of my research is to reach the point where I can make more sensible decisions for my patients. And that involves having a biomarker that tells me which of my patients are likely to respond to antifibrotic approaches. And one of the pieces of work we did there was to take sets of proteins that are showing potential in large proteomic discovery sets, one of which was MMP7. And working with this group, we're trying to get it towards the point where that and other proteins might help us develop a new diagnostic test. The first step was to confirm our reference material, our wild type and our knockout. And this shows that we've managed to do that fine. This work was led by Michael Biddle. And then we tested the same set of 10 antibodies, but only one of them was able to capture. And so we're trying to work with this company to see if we can start to use this data set as an asset towards developing robust diagnostics. This is only a platform for identifying antibodies with potential. There's lots of work yet to be done in this space. So this is my attempt to sound inspirational, but I have to say there's still an awful lot of work to do. But I guess it's close to 3 years now, we have managed to get to the point where we have a consensus protocol and agreed and published with those antibody manufacturing partners and a framework and international partnerships to test those antibodies and to try and get the best reagents into high quality science. Our work has led to one national funder announcing a new funding scheme and a review of their policy. We have had over 60% of reagent antibody manufacturers join the Seeker system. My colleague, Michael Biddle, has been awarded a Dorothy Bishop Award, which is our UKRN prize for open and reproducible research. We've published a case study to try and both highlight and sell the progress we've been making, but also to highlight the gaps and to get more engagement with the community to see how we can do this. We've had some funding and we've had some publications. So I guess I've probably, yeah, the timing's pretty good. So I guess my message is that progress is possible, but it's difficult. Working together to find shared wins where different stakeholders have shared incentives is probably the way forward. If you want people to engage with better practice, that better practice needs to be feasible, easy, and rewarded. And if you think you may have reasons for reaching out and collaborating, please do email me. I'll put that in the chat as well. And then there are just too many contributors to this work to thank than I have time to mention. I'm going to stop sharing and stop talking. Thank you so much, everybody, for your time.
Yeah, thank you so much, Have. I think we've realized that the conversations around open science and reproducibility have most of the time seem a bit abstract and always looking at publications in the end. So it's wonderful to see how this can translate into actual everyday practice, like the medical field, and how irreproducible, antibodies are affecting even patient diagnosis and etc., and how this could also affect patient outcomes. It's wonderful to hear the wonderful work you are doing. The sad part is the fact that there's not a lot of engagement for communities in the global south. That was the only sad part, and this is something you noted in your conversation and your eagerness to have people working with you. I don't know how has been the engagement with what the work you are doing right now from the research communities that you've engaged so far. How has been the engagement? Has there been enough buy-in, or a lot of people are still not aware?
So awareness is hard to measure because the tools we have to measure awareness tend to be self-selecting. So when we go to communities of early career researchers who feel safe to communicate openly and honestly, they say that this is one of the first times they've heard that this is even a problem, and that they wouldn't know how to go about solving this. And ultimately, we started where we started, which is in the UK, and obviously there's pros and cons to that, and we haven't even succeeded in reaching across Europe properly yet, let alone further afield. And ultimately, education awareness was really, really up there in terms of importance, and it's relatively feasible compared to expensive programs where we provide. So what we're trying to do is to reduce the work for others. There's always a resource problem, but part of the reason for wanting to speak to everyone here was you might have a early drug development program, you might be using antibodies. Email me, see if we can find a way of resourcing, finding the right tools for you to make your program more robust and more reliable.
Yeah. Thank you so much, Ab. Like you said, it's not easy to measure engagement, so a lot of work still to do in that regard. Yeah, so the floor is open for questions. You can just raise your hand or you can pop your question in the chat. It was a wonderful presentation from Harv and the work that YCharOS and Only Good Antibodies are doing. It can be a bit scary to know that this is what's happening in the medical field.
So I do need to clarify that I
Yeah,
mentioned early on there's diagnostics, there's therapeutics, and there's research use only. The regulatory frameworks around diagnostics and therapeutics are much higher than the regulatory frameworks around research use only, because so I don't want people to walk away with the impression that the drugs are dangerous or that the diagnostics are not reliable. It's mainly an opportunity cost. right
Yeah,
here and now, we could have more diagnostics, we could have better drugs, and we don't. And it's in part because of this inefficiency
yeah,
and irreproducibility.
yeah, yeah. That makes perfect sense. Research feeds into the outcomes of some of these things. And I mean, if research is being affected, then eventually, but like you said, the threshold in terms of the checks that are done at that level is quite higher. So we can be confident that what is being done out there is good. But on the same way, people like yourself who are involved in research will be concerned about some of these things that are happening because you
There,
know
there
that
there
eventually
are
we'll get
there
it.
are rare examples of things progressing that failed at a late stage because of this issue.
Yeah,
But I don't. This is another one of those things that it's always hard
yeah,
to critique science because science is the
yeah.
best thing that we have in terms of making progress. So if we start critiquing science too much, we don't want to turn people off science as an approach. And I struggle with this because science is still the best approach, but we could always do it better.
Yeah. Okay. So we have a question in the chat from Halata. The question is the technology can be applied in autoimmune diseases like multiple sclerosis?
Yep. So there's two elements there. So in autoimmune diseases such as MS, one of the things people are interested in is identifying antibodies inside the patient. And that is also a challenge to validate because you have to, is it really an autoantibody? Is it clinically important? And the systems you design for detecting them are like reverse ELISAs and they require tough validation. I'm aware of some platforms for discovering autoantibodies called CREC is an example. And but then you have a related but different piece of work to do to validate that your autoantibody detection assay is robust. Um, can my technology help you with discovering autoantibodies? I'm afraid my platform doesn't translate easily or quickly to that. But if Hala wants to reach out to me with a chat, I'd be happy to see what's possible. Maybe tell you the names of those platforms that I think are pretty useful.
Yeah. Thank you so much, Half. The floor is still open. You can raise your hand and then pop in a question, or you can just type it in the chat.
I
Uh,
think, Emmanuel, there's a question from Pauline asking. Pauline is a librarian at the Medical University Library, and she's asking how can her institution benefit? Like, how can her institution
Engage.
engage?
Okay. I'm trying to embed in the UK a champion for good practice
Mm.
in each institution. And what that looks like is we've put together a training program, and we're recruiting people, we're giving them some leadership, some technical training, but also some leadership training, because it's not just about the technical side, it's about being able to engage and influence. And I'm really open to exploring how that might go further afield. As a medical university librarian, I've also talked a lot about open data sharing because ultimately that Icarus platform is, I think, well, they've won prizes for their open data sharing because what they've done is they've taken a problem and turned it into an opportunity via open science, and they've made it safe for all of these different organizations to work together. And I think open data sharing is another thing that I think libraries often coordinate well, at least in my institution. So yeah, it's championing. So I'm just going to put my email in the chat, as well as some of the websites I mentioned.
Yeah, I think that would be really interesting, and I think this is something we could really help with if you're planning to expand your championship program, into Africa, because our community, 80% of it is from the biomedical field. And I think also 80% of them are academics, so they are probably working in research. So I think this is something that we could easily help spread the world about and let people know.
Very grateful and very open to those opportunities.
Yeah, great.
I think Nidal
Yeah.
has a question. Please go ahead, Nidal.
Yes, thanks for this informative and eye-opening talk. Actually, we also lead research groups on a multidisciplinary level for undergraduate and postgraduate researchers, and we are currently focusing in the awareness about because in Sudan during the war now there is a lot of infectious diseases happening there. So we are working in two sides in the raising awareness through patients and the community about the infectious diseases, and also in the medical fields or the medical community. So it can be applicable your platform or program to our groups so they can be aware of that. Because strategically, we are looking on these research groups who are working to be transformed into a business product that can be applicable and has impact in the community.
We're using a very similar approach in terms of ultimately, if we can find the right tools, we can realize value for businesses, values for health services, and ultimately value for society. For diagnostics for infectious diseases, I think, I started with COVID lateral flow tests as an example of when when the incentive is strong enough, people can find the right antibody, they can develop it into a diagnostic, a drug, and so on. And absolutely, I think we need to acknowledge that many diseases have been under-researched, under-resourced, and there are probably out there easy-ish opportunities to develop robust new diagnostics, maybe even drugs for understudied diseases. We've just not put the resources there yet. And reach out to me if you think you have a microbial protein that you think needs robust tools to be able to study. Our platform has centered on human proteins rather than microbial proteins. But our partners do include those who would be able to look at tools for microbial proteins too.
Okay, thank you.
That's really great. And I think thinking about, also me being a researcher in the medical field coming from Sudan, I think there are other issues that you might encounter in Africa. For example, our country, Sudan, has been under sanctions for like ages. And for us to get, the materials to do research has been really challenging. So we had to resort into, twisted ways to get these materials. So there's a missing link in the quality check of the materials, like you get an antibody, but then you don't know what happened in the process of this antibody,
The
like
cold
transporting it.
chain, yeah.
Exactly. So I think this would be a big thing that probably in Europe and in UK, this really wouldn't be an issue because you directly get the product from the manufacturer. But I think in Africa, this would really be an issue. It would. I could only imagine how much it would affect the reproducibility of the research that's been going on. So I was wondering if you're considering, things like that.
I attended a conference in Bath, in the UK just now called the Antibody Validation Conference. There was a company called Insilicate there who have developed a technology platform that reduced the need for cold chain for vaccines. And they are very interested in whether their technology, one of their missions is to be able to improve the ability for materials and reagents to safely reach everywhere. I don't, so that would be the thing that I'm most aware of, is that the solving that problem of the cold chain is on at least one company's agenda. And then the other thing is, I don't know the supply network across your region of the world. So, but I'd hope it's growing and improving, but I might be wrong.
Yeah, I think it's something that it would be really good to look into, since you already have contact with these manufacturers. It would be good to see like their customers base, like how many do they have in Africa? Like I don't know, because
I've spoken
yeah,
to friends in Saudi who say that there is a different and perhaps less availability of reagents to test, even in Saudi. So
yeah, then
clearly
it's
there,
worse than Africa.
so clearly there'll
Yeah.
be an issue, but
I think
technologies. Sorry.
I think, sorry for interrupting, but I think here in Saudi, the problem in a different context, that the regulations, the guidelines to do things, it is not well established yet. However, in compared to Africa, it's more worse because there is no even infrastructure to do research. But what I am seeing here in Kingdom, it's more applicable, and there is some higher authority they can listen. They can do actions when you rise up this problem or you have something to do.
Thank you. Emmanuel, you know that when you reached out to me with this, I worried the whether this would apply to everybody and whether
Yeah,
this would be helpful. And
yeah,
and I appreciate that perhaps sometimes it's not. I guess I hope that the approach whereby one, notice the problem, two, discuss it openly and honestly, three, reach out to those stakeholders who can influence things. And I'd say the other approach is to not take a blame approach. It's a case of, let's not worry about who is at fault, who caused this. How do we move forward? Like, and I think that probably applies to a lot of reproducibility and research integrity problems.
yeah. And I
Yeah.
think you were talking earlier that in your engagement, you've encountered a lot of people that were not aware of the problem. And when they became aware, the question was, how do they solve it? And I think it's good to have you currently available as a contact to get, let's say, knowledge or information about how to support about some of these things. Like Lame said, I'm sure it's a problem that is cutting across a lot of African countries, and not only in Africa, a lot of global South countries. I think a lot of people would be interested in engaging on this thing. I think someone just posted something. Tracy, wonderful.
So the next generation sequencing reagents, CDC Africa is facilitating. Yeah, I guess I don't know the need in different parts of Africa for either research grade antibodies or diagnostic grade antibodies. And it'd be interesting if, I mean, Tracy, if you did reach out to me to understand what is the need. Do you know? Drug development is happening in different places, and some of those will be in relation to human targets. Other times there's infectious diseases. Again, this issue of studying proteins is very broadly relevant across species, diseases, and yeah. I I will definitely reach out once we've put together our educational material. Everything will be shared openly and freely. The whole Champions program too.
Yeah,
And I'll reach out to both of you with the links, etc.
yeah, definitely. I think Tracy has added something extra. Okay. I think yeah, Tracy, could you unmute yourself and talk that we could get a better perspective from you.
Hey, yeah, if you
Yeah,
can hear me, I didn't see
yeah, we
also.
can.
Okay. Yeah, I know that the Africa CDC is doing a lot of work in trying to get some agents to, and that already is a complicated process. But as far as I know, they've done it predominantly for NGS reagents. In terms of antibodies, as Avendra said, I think that is mostly done on an academic level where people are buying these for academic research. I don't know to what extent that is happening in the public health research. Um, and then drug development studies, I really don't think I know many manufacturers here in Africa. I know that drug development in the context of protein modeling for bioinformatics is growing and like really growing substantially, but I don't know yet because I know Kenya even recently lost their ability to do some manufacturing for COVID. So I don't know how much. Maybe that's a separate issue. There needs to be more trust for things like this to happen on the African continent, but that's a separate issue.
Nonetheless, Tracy, I think you have brought up an important issue, which is that informatics studies may at some point touch data that depends on antibodies. And it's so, for example, some proteomic platforms, Olink. So if you're, it's not easy to know that the underlying data will be robust unless you're aware of this. And if you're using open data for your research, it might be helpful to know where antibodies were used and what the pros and cons and how to look at that data to understand whether the antibody might be robust or not. Ultimately, genomics data doesn't use antibodies, and I assume some of that will have been genomics data.
Yeah, thank you so much, Av. I think we've exceeded 1 hour. I don't know if there are any other questions. If there are any question, you can raise your hand or you can pop it in the chat. If not, I think it's good that Tracy mentioned the Africa CDC. I think the work you're doing is so important, and you have 60% of the manufacturers of these antibodies and this product. And I think it would be good to maybe reach out to some of these groups like the Africa CDC and organizations like that. I think it would help with your engagement and make it more streamlined, because I think they have connections with some of these groups, the medical groups and researchers that are in some of these practices. So it will really make your work a lot easier. I don't know if Tracy is connected to them and knows someone there, but like you said, she can reach out and you can see how you can work together on this. I know Tracy from our work together in the African bioinformatics community, and I think it's wonderful to have a perspective concerning this side and to see how you can work around it. Like you said, definitely informatics data could touch on some of these things because it definitely comes out of research, and if the downstream processes are not really well done, it could also affect the outcome. So it's wonderful to have people like that here. Are there any other questions, or do you have any other things that you'd want to mention?
No, so thank you so much, Emmanuel, Lemise, and all of your attendees for listening in. Please do reach out, and I will pass on educational resources, free educational resources, as and when we're ready to release them.
Yeah, sure. I think I saw Hala raise the hand, but I don't know if that was a mistake. Hala, did you want to talk? Yeah. Hala, you can unmute yourself and talk. Yeah.
Yes, okay. Thank you for this very insightful session. Okay, as the Dr. Lemise has indicated that at this time there are a lot of epidemics and outbreaks in Sudan, especially after the war. We have a huge dengue fever outbreak, and the remaining research institutions that could resume their research, they're just researching or trying to find a way to do research on the dengue fever now, and there are a lot of hypotheses talking about proteins in the immune system that if it have been blocked by the virus, the treatment is not efficient. So they are talking about using antibodies and so. I don't know how can we reach or how can to get the benefit of using antibodies in researching dengue fever in this time in Sudan.
Okay, so it sounds like some component of the immune system is affected by the dengue virus, and robust tools to be able to study that component of the immune system.
Yes, exactly.
Yeah.
Exactly. Yeah.
You'll understand that if I was answering that question in my setting, that would be a case of a flow cytometry assay to be able to determine which immune markers express the protein of interest, and then using a validated antibody, validated perhaps through the process that I've outlined in my presentation, to be able to I'd call it immunophenotype on the basis of that marker to say, if the question was, does this marker affect susceptibility to dengue fever and clearance of dengue fever? That's a very common question for immunologists, and that's a very common approach that people might take. I don't know the specific protein you're talking about, and I certainly don't know or expect that there would be the ability to run a study like that in Sudan at this moment in time. But at the same time, dengue fever is not Sudan specific. There are other places where that question, but I get it's not fair that Sudan shouldn't get to contribute to answering that question. Yeah.
Yeah, Nidal.
Yes, unfortunately, it is also linked with co-infection with malaria. So this is the situation: co-infection, dengue fever, and malaria.
Okay.
Thank you.
Yeah. Reach out if you have the specific papers and hypothesis, and yeah, very eager to be connected to the people and see what we can do to help them. And for example, there are foundations that could help us resource work. And you know, there are a number of funders who, the Gates Foundation, the Wellcome Trust, who understand that we're not necessarily looking at global needs when we're working here in the UK. We should make sure that we take the global need into account when we design programs, and our work could help facilitate programs.
Okay, that will be great. I will definitely contact you. Thank you so much.
Thank you.
I will. Thank you.
Right. I think I see Nidal.
Yes. Yeah, can you hear me?
Yeah.
Okay, so I have this question about individualized treatment and personalization, where the antibiotic treatment we're using as a diagnostic will be combined or fill-in will be used in terms of individualized treatment and personalization.
So a diagnostic marker for individualized treatment for infections as well as the duration of treatment, is that correct?
In also in the immunological disorders or diseases, how it will be used.
My perception of how individualized, I'm not an infectious disease specialist, and although
No,
I'm
I mean
aware,
even for the immunodiseases, how it can be used in the individualized
okay,
treatment.
so for us, what that
Yeah.
looks like is we have cohorts of patients who we have done proteomic studies using mass spectroscopy, and we have identified proteins that we think might predict disease progression, i.e., people with this high level of this protein progress faster. We develop robust tools to measure that, and we try and develop those tools such that they're cheap and scalable. Even here in the UK, if you want to have a blood test, if it costs more than 50 pence, which the NHS is not interested, so we have to develop assays that are scalable, and our technology partners are potentially in a position to start to do that, and that's the approach. It's identify a marker that predicts an outcome, then develop a tool that can measure it in a scalable inexpensive way.
Okay, thank you. I really like the ecosystems that you built. Thank you a lot.
Thank you.
Yeah, thank you so much, Hav. I think we are almost 10 minutes past our time. I don't know if you still have a few minutes, or we can just close the session. I think the questions. Okay, someone has posted a question. Tracy, yeah, Tracy has posted something.
Some information.
Yeah. Okay. I think it's a response to the.
Yep.
Yeah. That's good. Yeah, that's good. Thank you so much, Tracy, for pointing out to this one. Yeah, thank you so much. I think I'm not sure there are any other questions now. Yeah, thank you so much, Hal, for this session. I think it was very insightful and it was exciting. And I was sad that we are having this session for ourselves and we are benefiting it alone. And I'm like, can't we have to speak with a lot more people across Africa? Because this is wonderful work that we want a lot of people to know about. So it's wonderful that you are doing the Champions program, and would really love to engage on it and how we can make a lot of people aware.
Very grateful for you guys giving me the time.
Yeah. Thank you so much, Hava. And thank you everyone for joining us today. The questions and the input and the discussion was so wonderful as usual. And look out for emails coming from the team about our next session. Thank you so much, Hava. And
thank you.
We look forward to engaging you further. Yeah.
Take care.
Have a wonderful day.
Thank you. Bye-bye.
Yeah.